Welcome to today's custom-tailored deep dive. You, the listener, have actually given us a really fascinating, honestly pretty dense stack of high-level sources to open up today.
Oh, definitely dense. I mean, we are looking at some very comprehensive reports from the CDC and the WHO, alongside some incredibly cutting-edge research reviews from journals like Lancet Microbe and Cell Host and Microbe.
Right, heavy hitters. And the topic on the table is this silent, escalating global crisis of antimicrobial resistance, or AMR, as you'll hear it called.
Exactly.
So our mission for you today is to cut through all that heavy medical jargon, shortcut your path to understanding exactly how humanity lost the upper hand against bacteria, and then, explore the sci-fi level technology we were deploying to win it back.
It really does sound like sci-fi once we get into the newer tech.
It totally does. But to really understand how fragile our medical arsenal is and how high the stakes are right now, I want to take you back to 1941, to a hospital in Oxford, England, and a police officer named Albert Alexander.
Yeah, the Albert Alexander story, it's tragic, really. He had what started as a minor scratch on his face, maybe from a rose thorn in his garden.
Just a rose thorn.
Right. But it developed into a severe, highly aggressive bacterial infection. It spread to his scalp. His eye actually had to be removed, and, well, he was dying. Conventional treatments of the era were completely failing him.
But researchers Howard Florey and Ernst Chain had this experimental new compound, right? Derived from a specific mold.
Yes.
No.
They called it penicillin. And with no other options on the table, Albert Alexander became the very first human to be treated with it.
And the initial results must have felt like absolute magic to those doctors. I mean, within days of receiving the penicillin, his fever plummeted, his appetite returned, and this horrific infection actually started clearing up.
The drug was doing the impossible.
Yeah. But the heartbreaking twist here, which really sets up everything we're unpacking today, is the sheer physical difficulty of producing that drug in 1941. They simply didn't have enough of it.
No, they didn't. The extraction process was painfully inefficient at the time. I mean, the researchers were literally growing the mold in whatever vessels they could find. They were using hospital bedpans.
Wait, bedpans? Seriously?
Literally bedpans. Just trying to glean a few precious milligrams of the active compound. So when Albert's supply began to run low, the medical team had to resort to a staggering measure of desperation.
This is the part that just blows my mind.
Because penicillin is excreted by the body relatively quickly,
Yeah.
doctors collected Albert's urine, chemically extracted the unmetabolized penicillin out of it, purified it, and re-injected it back into him just to stretch the treatment out.
It's an unbelievable image. Filtering a man's urine just to squeeze out one more dose of a miracle.
Yeah, but ultimately, even recycling the drug like that wasn't enough. The supply ran dry.
The bacteria surged back, and Albert Alexander died.
He did.
It's a profound reminder of how precarious our chemical weapons against disease have always been. And, when you hear a story like that, it's easy to assume that antibiotics are this purely modern human invention, and that bacterial resistance is just a modern human glitch like a mistake we made by overprescribing pills in the 20th century.
It is a super common assumption, but diving into the Cell Host and Microbe review, it dispels that myth completely. Antimicrobial resistance is not a human invention at all.
Right.
It is a 3.4 billion-year-old evolutionary feature of this planet.
3.4 billion.
Yeah. Genomic studies show that bacterial resistance to beta-lactams, which is the specific class of antibiotics that penicillin belongs to, that resistance is roughly 2 billion years old. Resistance to other classes, like glycopeptides, dates back 500 million years.
Wow. So all of this was happening long, long before humans even walked the earth.
Exactly. We really have to visualize what was happening in the soil for those eons. It was a microscopic, micron-level war. Fungi and bacteria were constantly competing for the exact same resources in the dirt.
So a fungus would randomly evolve a toxic chemical and antibiotic to kill off the competing bacteria, right?
Yes, and in response, the surviving bacteria would evolve a genetic defense mechanism to neutralize that specific chemical. It has been a relentless, invisible arms race.
When you look at it through that ecological lens, the medicines we use today aren't really our inventions. It's like we just stumbled upon these ancient pre-existing weapons.
We borrowed them from environmental fungi and microbes that have been fighting each other since the dawn of life.
Right. It's like finding a cache of stolen alien weapons, figuring out how to pull the trigger, and then thinking we invented the laser.
That's a great way to put it. And the historical clues were always there, warning us The sources mention Paul Ehrlich, one of the pioneers of early antimicrobials. He actually predicted bacterial resistance to his arsenic-based treatments way back in 1907.
Even before penicillin.
Exactly. And even more startling, in 1940, before penicillin was even widely mass-produced for the public, scientists Abraham and Chain discovered an enzyme in an E. coli bacteria that could actively destroy penicillin.
So the defense mechanisms were already pre-installed in the bacterial genome, just waiting to be activated.
Always waiting.
Okay, so if AMR is this ancient, perfectly natural phenomenon that has been happening in the soil for billions of years, why is the CDC suddenly labeling it an urgent global public health threat right now? What tipped the scales from a microscopic dirt war to a modern crisis?
Well, the tipping point is the sheer scale and speed of human behavior. We took this localized microscopic ecosystem and we supercharged it. We extracted those weapons, mass-produced them, and flooded our hospitals, our agricultural systems, and our waterways with them.
We just drenched the planet in them.
Exactly. And by doing that, we put unprecedented evolutionary pressure on every bacteria on Earth simultaneously. The data from 2019 shows AMR was directly responsible for 1.27 million global deaths.
1.27 million deaths
Yeah.
just in 1 year.
Yes. And if the trajectory doesn't shift, the projected global economic loss by 2050 is staggering. They're estimating somewhere between $100 and $200 trillion.
Those numbers are almost too big to comprehend. And the sources point out that managing this threat requires a massive paradigm shift, something called the One Health concept.
Right, One Health. It's the recognition that human health does not exist in a vacuum. It is fundamentally intertwined with animal health and the wider environment.
Because you can't solve antimicrobial resistance simply by telling family doctors to write fewer prescriptions for human sinus infections.
No, you have to look at the entire global ecosystem. Take the United States in 2021 as an example. 54% of all medically important antimicrobials sold weren't given to human patients. They were used in agriculture, administered to livestock.
And often given at subtherapeutic doses, right? Meaning they aren't even treating a sick animal, they're just mixing a low dose into the feed to promote growth or prevent illness in these really crowded conditions.
Exactly. It's a selective pressure cooker.
But looking at that 54% figure, my immediate reaction as a learner is, why not just ban agricultural use globally overnight? If farming is driving half the problem, just cut the cord.
A sudden global ban seems like the most logical lever to pull, but the reality of global food security makes that incredibly dangerous.
Really? How so?
Well, in many lower and middle-income countries, the economic survival of small farmers and the availability of affordable protein for entire populations rely heavily on these drugs. Livestock in these regions are often raised in less than ideal sanitary conditions.
Oh, I see.
So stripping away antibiotics overnight would trigger massive die-offs, food shortages, and economic collapse for vulnerable populations.
So it's not just a lack of willpower to fix this, it's a structural trap.
Right, it's highly complex. That's why the WHO advocates for a coordinated phase-out rather than a catastrophic ban. It involves improving animal hygiene infrastructure, investing heavily in veterinary vaccines so the animals don't get sick in the first place, and slowly stepping down the reliance on growth-promoting antibiotics.
Okay, that makes sense. But if agricultural pressures are actively training these bacteria to survive, what happens when those newly trained microbes collide with a global crisis?
That's where things get really bad. The sources show how these environmental pressures bleed directly into our medical systems.
Yeah, the COVID-19 pandemic is a perfect example in the sources. Early in the pandemic, before we fully understood the virus, there was a drastic increase in inappropriate antibiotic prescribing.
People were panicking.
Right. Add in the fact that severely ill patients were kept on ventilators and catheters for prolonged periods, creating the absolute perfect high-stress breeding grounds for resistant hospital bugs.
And then you throw climate change into the mix. With rising global temperatures actively accelerating the evolution of heat-resistant pathogens, we have a perfect storm. We are forcing environmental bacteria and human pathogens into the same high-stress arenas,
causing them to interact and share survival strategies in ways they never have before. Which opens up a huge question about the mechanisms at play here. Like, how exactly are these bugs surviving our best drugs? Opening up the bacterial playbook in these sources is fascinating because, honestly, bacteria are incredibly social creatures when it comes to survival.
They really are. They share genetic cheat codes with each other through a process called horizontal gene transfer.
Horizontal gene transfer. Break that down for us.
So instead of just passing traits down vertically from a parent cell to an offspring cell during division, bacteria have the ability to pass resistance genes sideways, often to entirely different species of bacteria.
That's wild.
Yeah, and they accomplish the sideways transfer through three primary methods. The first is transformation, where a bacterium literally slurps up free-floating DNA left behind by dead neighbors in its environment.
Slurping up dead neighbor DNA,
Yes.
but effective.
Very. The second is transduction. This is where a bacteriophage, which is a virus that infects bacteria, accidentally packages some bacterial DNA from one host and injects it into the next bacterium it attacks.
And then the third method, conjugation, which is essentially bacterial sex, right?
Essentially, yes.
Two bacteria sidle up to each other, form a tiny physical bridge or tube between their cell walls, and directly pass genetic material across. And the material they are passing is often packaged on mobile genetic elements, like plasmids.
All right.
Plasmid is basically a biological USB drive. One bug hands the other a USB drive containing the exact step-by-step blueprints for surviving a penicillin attack.
And once a bacterium downloads those blueprints, it physically rebuilds itself. The research highlights four main physical defenses they construct. The first is decreased permeability.
Decreased permeability, so keeping the drug out entirely.
Exactly. Bacteria have tiny pores on their outer membranes called porin channels, which let nutrients in. When an antibiotic tries to use those same channels to enter, the bacterium simply alters or closes the pores. The drug bounces right off the outer wall.
But if the drug does manage to get inside, they use the second defense target modification.
Right.
I like to think of the antibiotic molecule like a key designed to fit exactly into a specific bacterial protein lock to shut the cell down. The bacterium just subtly mutates the shape of its lock. So the drug is still floating around inside the cell, but it can't turn the mechanism anymore. It's totally useless.
It is. And the third defense is far more aggressive. Antibiotic inactivation. Instead of just hiding the target, the bacterium produces specific enzymes to actively hunt down the drug.
The sources mentioned beta-lactamases and the NDM enzyme for this.
Yes. These enzymes act like microscopic scissors. They physically bind to the antibiotic molecule and snip its core chemical ring, chopping it to pieces before it can harm the cell.
Okay, looking at the fourth defense, efflux pumps, I'm just picturing microscopic bouncers at a nightclub.
So it's exactly what they are.
Right. The antibiotic molecule manages to sneak past the front door, gets inside the cell, and before it can do any damage, this protein pump grabs it by the collar and physically tosses it right back out into the street.
And the terrifying part about those cellular bouncers is that they aren't checking IDs. Some of these efflux pumps are non-specific. They don't just throw out a single type of drug, like penicillin. They will grab multiple, entirely different classes of antibiotics and pump them all out.
So they're completely multi-drug-resistant.
Yes. And this combination of genetic sharing and multi-layered physical defenses has led to the rise of the escapee pathogens.
Escapee, spelled with two E's at the end.
Right. It's an acronym for a group of highly virulent, multi-drug-resistant superbugs that are increasingly responsible for deadly, untreatable, hospital-acquired infections worldwide.
Man, if these escapee bugs can share resistance via plasmids, mutate their targets, use microscopic scissors to chop up drugs, and use efflux pumps to literally throw multiple medications out the door, it really sounds like chemical antibiotics are fundamentally a dead end. Is the WHO just telling doctors to prescribe less and cross their fingers? Or is there an actual scientific pivot happening here?
Well, the immediate reaction is a stalling tactic. The WHO implemented the AWARE classification system, sorting antibiotics into access, watch, and reserve categories.
Okay.
It is a strict management guideline for clinicians. The goal is to ensure our absolute strongest, last resort antibiotics, the reserve category, are kept locked in a metaphorical glass case. You only break the glass when a patient is facing a completely resistant infection. This prevents bacteria from getting early, low-dose exposure and developing resistance to our final lines of defense.
But rationing a dwindling supply just buys us time, doesn't solve the problem.
No, it doesn't.
The real excitement in these journals is the pivot away from static chemicals toward radical biological alternatives. We are talking about deploying entirely new mechanisms, for instance, antimicrobial peptides.
Yes, peptides.
These are naturally occurring molecules with names like LL37 or omeganin that operate differently than traditional drugs. They don't just poison internal cellular machinery, they physically integrate into and shatter the bacterial cell wall.
They're devastating to the bacteria, and the research on CRISPR-Cas9 gene editing is equally profound.
Oh, CRISPR, I love this part.
Instead of trying to kill the entire bacterium with a chemical, researchers are using CRISPR as a microscopic genetic scalpel. They can program it to enter the bacterium, hunt down specific resistant sequences like the MCR1 gene, which provides resistance to the highly toxic last resort drug colistin, and literally snip that specific gene out of the bacterial DNA.
So you aren't killing the bug directly. You are reprogramming it to make it vulnerable to normal antibiotics again.
Exactly.
And then we have bacteriophages, which sounds straight out of a sci-fi novel. These are viruses that have naturally evolved in the environment to do one thing: hunt, infect, and kill very specific types of bacteria while leaving human tissue completely untouched.
They're highly specialized assassins.
But exploring this raises a major question for me. Aren't we just fighting fire with fire? I mean, if bacteria spend billions of years evolving to resist chemical antibiotics, won't they just use their plasmids and efflux pumps to evolve resistance to these bacteriophage viruses, leaving us exactly where we started?
It's a great question. And yes, bacteria absolutely will evolve to evade phages. The fundamental difference, though, lies in the nature of the weapon itself. Traditional antibiotics are static chemicals. Once a bacterium mutates its lock to evade a chemical key, that chemical is permanently useless.
Right.
But a bacteriophage is a biological entity. It is alive, and it has its own DNA. When the bacterium mutates to evade the virus mutates right back to catch the bacterium.
Oh, wow.
This is coevolution. It keeps the arms race dynamic and competitive, rather than humanity simply losing outright to static drug resistance.
The medicine actually adapts in real time alongside the disease. That is incredible.
It really is.
But, finding the exact right bacteriophage for a specific patient's infection, or designing a custom CRISPR therapy, that takes immense time, money, and laboratory resources. And time is the one variable Albert Alexander didn't have. And it's the one we don't have now.
Time is definitely the enemy here.
Right. To truly jump ahead of bacterial evolution, we can't spend decades searching for the next mold in a petri dish. We need to completely reimagine how we discover treatments at lightning speed.
And this is where artificial intelligence and machine learning are completely revolutionizing the landscape. The speed bottleneck starts at the very first step, which is diagnosis.
Right. Because currently, if a patient presents with a severe infection, doctors take a blood or tissue culture and have to wait 24 to 48 hours for the bacteria to physically grow in a lab before they can test which drug kills it.
It takes days. And during those crucial 2 days, doctors are forced to guess, often prescribing broad-spectrum antibiotics that blindly wipe out good and bad bacteria alike, which of course actively contributes to the resistance problem.
But the Lancet Microbe paper outlines a massive leap forward here. AI-powered nanomotion platforms.
Yes.
Instead of waiting days for bacteria to multiply, this platform uses artificial intelligence to measure the microscopic physical vibrations of the bacterial cells in real time. If you apply an antibiotic to the sample and the tiny vibrations stop, the bacteria are dead. This technology can determine the exact right targeted antibiotic to use in just 2 to 4 hours.
The guessing game is eliminated.
Completely eliminated.
And beyond diagnostics, AI's role in the actual discovery of new drugs is just staggering. Historically, discovering a novel antibiotic meant painstaking, manual trial and error by human scientists. Recently, researchers unleashed machine learning algorithms to scan over 60,000 environmental metagenomes.
A metagenome being essentially a massive complex soup of genetic data scooped right out of an environment like soil or ocean water, containing the DNA of thousands of different microscopic organisms all mixed together.
Exactly, and manually sorting through a single metagenome would take a human researcher years. But the AI sifted through 60,000 of them in a fraction of the time. It identified nearly 1 million potential new antimicrobial compounds hidden in that genetic soup, including highly promising ones like lacnosprin-1.
Finding hidden natural weapons is one thing, but generative AI pushes this into entirely uncharted territory. We are used to seeing AI generate images or write text, but platforms like Synthamol are virtually generating entirely new molecular structures.
It's mind-blowing.
It is. The platform virtually combined different molecular fragments to generate 30 billion entirely new virtual molecules.
Yeah.
30 billion. It checked those billions of molecules to ensure they wouldn't be toxic to human cells, and then provided the chemical instructions on how to physically synthesize them in the real world. This process has already led to the discovery of completely structurally unique drugs like halicin and abaucin.
And the significance of AI-generated drugs like halicin and abscin is revolutionary because they do not look like any antibiotic found in nature. They operate on entirely novel mechanisms of action. And in lab settings, they have proven capable of killing completely dormant, highly resistant strains of bacteria that brush off our strongest traditional drugs.
Thinking about generative AI here, it is like having a master chef creating recipes using chemical ingredients that bacteria have never even tasted.
I love that analogy.
Because these molecular structures are completely synthetic and entirely new to the planet, the bacteria don't have those pre-existing ancient genetic defenses waiting in their DNA to fight them off. The bouncers don't know what to look for. The microscopic scissors don't fit around the molecule. We are finally finding a way to bypass the 3 billion-year-old cheat codes.
We are leveraging the sheer computational power of the 21st century to outpace the evolutionary power of the microbe. But a crucial takeaway for any learner diving into these sources is that fighting AMR isn't just about passively waiting for an AI superdrug to hit the pharmacy.
Definitely not.
It remains a globally connected One Health challenge. Every single time an antibiotic is flushed down a drain or utilized unnecessarily in an agricultural feedlot or demanded by a patient for a viral cold that it cannot cure, we are actively contributing to the evolutionary training of these pathogens.
We really are all in this together. And, we have covered an incredible expanse of ground today. We started by reframing our understanding of a 3 billion-year-old microscopic war playing out in the dirt. We looked at the heartbreaking scarcity of penicillin and the desperate attempts to save Albert Alexander in 1941. We unpacked the complex bacterial playbook from sharing USB drive plasmids to deploying efflux pumps.
It's a massive playbook.
And we ended up exploring a future where biological phages co-evolve with infections and generative AI invents synthetic molecules to save our lives. It is a brilliant testament to human ingenuity, but also to the relentless adaptive power of nature.
It is a highly precarious balance. The sources demonstrate that we possess the technological and analytical tools to manage this crisis. However, executing that management requires unprecedented global cooperation, strict stewardship of the chemical drugs we currently have, and massive, sustained investment in the biological and computational alternatives of the future.
Well said. Before we wrap up this deep dive, I want to leave you, the listener, with a final provocative thought to mull over. Our sources made it abundantly clear that highly confined environments, physical stress, and the intimate mixing of human and environmental microbiomes drastically accelerate the horizontal gene transfer of resistance.
They do.
Now, look to the near future. As humanity prepares for a multi-year spaceflight to Mars, think about what happens to the human microbiome inside that ship.
Oh, wow. When you apply the variables of AMR to a multi-year spaceflight, it becomes a fascinating and potentially terrifying equation.
Yeah. Right. You have a sealed, highly confined capsule where nothing enters or leaves.
Yep.
You have astronauts under extreme psychological and physiological stress. You're constantly recycling the same water and air through the system for years, and the entire environment is being bombarded by elevated levels of cosmic radiation, which is known to cause rapid DNA mutations. Are we inadvertently designing the absolute perfect high-pressure incubator for the next generation of superbugs?
That is a chilling thought.
And if a resistant infection breaks out halfway to Mars, well, we might find ourselves right back in a hospital bed in 1941, hoping against hope that the medicine doesn't run out. Keep questioning, keep learning, and we'll catch you on the next Deep Dive.